All right, everybody, good morning and welcome to Micro Seminar. I'm here with Nick Peng at the University of California, Santa Barbara, my home institution. I'm Lizzie Wilbanks, and I'm really happy to have Nick here with me today from across campus, able to share with you guys some of his exciting new work on fungi. So Nick did his graduate work at Princeton University with Brett Ward, looking at nitrogen cycling under different redox environments and the effect that that has. In the last 2 years, he's been a postdoctoral fellow here at UCSB with Michelle O'Malley and Dave Valentine, looking at all things fungal. We're going to get to hear about some of his exciting projects here today. So without further ado, I'm going to turn it over to Nick. For those of you who are watching the broadcast or following us on Twitter, go ahead and tweet us your questions during the talk. Nick would be happy to take them at any time. So,
all right.
Over to you.
Thanks, Lizzie, for the kind introduction. I'm going to start a screen share. All right, so let me take care of this hide. Okay, good morning, everyone. Thanks for tuning in. And I'd apologize ahead of time if I have to cough or blow my nose because still recovering from a cold. And today I'm very happy to share with all of you some of the work I've been doing since I started my postdoc at UC Santa Barbara. And the title, as you can see, spans from the anaerobic fungi to marine fungi. So the overarching question as a biogeochemist that I always pursue is to understand the interactions between the environment and the associated microbial communities, which mediate biogeochemical cycles of elements such as carbon, nitrogen, sulfur, etc. And among these environmental parameters, we can consider, typical ones such as the physical parameters, including temperature and light, the chemical ones such as the nutrient condition, macronutrients, micronutrients, pH, etc. And in addition to that, it's also important and has been drawn more and more attention in the past decade, which is the interactions among microorganisms themselves and how that would have an effect on microbial community. And for today's talk, I have a simple outline breaking them down into first a part of the talk talking about anaerobic fungi, which is what I've been primarily working on as a postdoc since I came to Santa Barbara. And then very recently, since a few months ago, I've started also working on marine fungi. So this is just an attempt of me trying to draw a cow, but the point here is just to show the gut microbiome of cow include a number of different microbes from different taxa, in fact, different domains, including bacteria, archaea, protozoa. And these microorganisms are symbiotic with the cow or any herbivorous animals. So they, in fact, we don't know any herbivorous animals that possess the gene or the enzyme to degrade cellulose and hemicellulose, which is a major part of their diet. So they have to rely on their gut microbiome to degrade these cellulose and hemicellulose. And the major products of these microbial degradation of biomass rich in cellulose and hemicellulose include volatile fatty acids, ethanol, and also hydrogen sometimes, and methane. And we know as a fact that the anthropogenic driven consumption of beef has a very big impact on the methane production because we're raising a lot of cattle and they are actually producing a large amount of methane. And if we look at the abundance of different microbes in the gut microbiome, generally bacteria, like in most other environments, outnumber their eukaryotic counterparts by several orders of magnitude. So bacteria could be about 10 to the 10th per gram of rumen fluid, whereas protozoa is only about 10 to the 6th per gram of rumen fluid. So anaerobic fungi, they really have been observed as early as 1910 in the digestive tract of herbivorous animals. But they have long been overlooked. And this is going to be a theme of what overarching theme that I'll be talking about today, which is these fungi are really overlooked in these environments. And they actually have been misidentified as ciliates for over half a century. So it was only in the 70s when Colin Orpin, who did a lot of the early work in anaerobic fungi, that they discovered these anaerobic fungi contain cell walls made of chitin, and they truly belong to the fungal kingdom. So just a quick plug about the potential of these anaerobic fungi. So they could potentially be used for converting biomass into biofuel if we consider using them with the other microorganisms that are native to the guts of herbivores. And a quick fact about a lot of developing countries, which is that a lot of crop waste in developing countries are simply burned as a common practice. Take China as an example. There is about 100 to 150 teragrams of crop waste that are burned every year. And that could contribute to as much as 5% of the total emission. And imagine that if we can channel those carbon actually through fungi and other microorganisms, we can potentially generate biofuel and reduce and better harvest the energy before those carbon are released into the atmosphere. So how do we get to these anaerobic fungi? But when I came to Santa Barbara, I was very lucky to work with Michael Theodoro from the UK. And he has been working on the anaerobic fungi for over 30 years. And this is him pictured in the photo on the left, collecting giraffe fecal pellets at the Santa Barbara Zoo. And he is also the one who developed some of the very important techniques in growing and maintaining anaerobic fungal cultures. And on the right is a different sampling area in the Santa Barbara Zoo. And what we do to get these source microbiome is that we actually wait outside of this enclosed areas of sheep and goats and watch for fresh production. And as soon as the droppings comes out, we would run up and then put the fecal pellets into the 50 ml centrifuge tubes we brought with us. And then immediately we bring these back to the lab and start a enrichment culture. So you can see that from these goat fecal pellets, we would grow them anaerobically, either in fungi tubes or in serum bottles in a complex media that is supplemented with rumen fluid. And this is one of the major challenge that we're still facing is to design a reliable, defined media that does not rely on rumen fluid as a supplement to these media. But in order to really get these microorganisms to grow, we found that 90% of the time we have to rely on the rumen fluid supplement. And what you're seeing here on the left is a fungi tube with some wreath canary grass in them, and then it's already floating to the top. On the right is a bottle of media with these are just filter paper as a carbon substrate. And on the bottom is how we isolate these anaerobic fungi. So after they are growing in these liquid media, we take a small amount of them and inoculate them into these row tubes. And the row tube is basically just an anaerobic agar plate, but in a three-dimensional surface. And you can see these dots in this row tube. These are individual colonies of anaerobic fungi. And as you can see from this photo, well, it's not easy to see it well, but these anaerobic fungi, when they grow, they do not really increase the turbidity of the culture. So how can we evaluate non-invasively the growth condition and how fast they're growing and which stage they're at of these anaerobic fungi? So one way that was developed by Mike Theodoro is this pressure transducer method. So basically, these fungi, they produce a large amount of carbon dioxide and hydrogen in the headspace. So we simply measure the amount of pressure accumulated in the headspace between a certain time period and then add them up. And this will give us a very good approximation of the growth curve of these anaerobic fungi. And this has been validated with by measuring their biomass at each time point. And below here is one of the growth curve I have generated coming here. So they have generally a couple of days of lag phase before they go into the exponential phase, and then they enter the stationary phase after about 4 to 5 days. So these anaerobic fungi don't grow very fast. And on the right is just another very short video I took after with atmospheric pressure.
Nick, can you repeat what you just said there? We had a little trouble hearing you.
Oh, okay. What I was saying is that, so for what I'm showing here is just a quick video of a bottle of anaerobic fungi culture, and just showing that when, okay, I see. Yeah, the video actually had some sound to it. So yeah, okay. Yeah, basically after we measure the pressure in the headspace, we release the pressure and equilibrate it with atmospheric pressure. And all these graphs, as you can see in this photo, that were at the bottom of the culture, all floats up because of the release of pressure. And you can also see that they sort of form a mat of grass, whereas the grass at the beginning were in fact were all loose. So what did these anaerobic fungi really look like, right? So I'm going to show you a few photos that I took, and this is, so if we go back here and look at this fungi tube, we can actually just put this under an inverted microscope and observe directly of what is in the culture. So what you see here in the middle, the dark part is a piece of plant particle. And on top of it, these three bulging cells, they are the anaerobic fungi, probably a developing sporangia. And the error bars are 10 micrometer. And if we look directly into the row tubes, you can see that there are a lot of these anaerobic fungi swarming towards some carbon source in this row tube, because the carbon was not really evenly distributed in the row tube in this case. And they contain all these flagella behind them, which provides them with mobility. So what happens in the rumen is that they actually have the capability to swim towards a biomass or plant substrate. And if I take just a small drop of the liquid culture onto a glass slide, I can get a better image of them close by. So these are their baby form, zoospores, and these are also what was confused or misidentified back in the early 1900s as the ciliates. And as you can see that they have multiple flagella in this case, and they're very round on the top. And if you can see them moving under a microscope, they generally try to swim towards some carbon substrate. And when they grow larger, you can see that they will grow into these larger sporangia with very sophisticated rhizoidal structure. And this is actually also something that distinguishes them from their bacterial counterparts in the gut microbiome, because these rhizoidal systems are very strong and they physically break down the plant biomass and hence creates more surface area for all other microorganisms in the community to access the biomass. And on the bottom is just a different form of these rhizomycelium. So I believe on the top, what we're seeing are these rhizoid system that are nucleated. So they're thicker. And on the bottom, these rhizoidal systems, they are non-nucleated. So this brings us to a very brief view of the life cycle of anaerobic fungi. And this is taken from a very nice review written by Gruningers and colleagues. So we can start basically from this phase. So these on the left side are zoospores. And you have seen a few photos of them in the previous slide. So they would swim around in the culture or in the gut towards plant material. And once they have established themselves on a piece of plant material, they will start to turn into their vegetative state and germinate from cyst. And over the time, develop a rhizoidal system that you have just seen. And for the monocentric fungi, the nucleus remains in the cyst, in the round part, and they eventually develop into a mature sporangium. Whereas for polycentric anaerobic fungi, the nuclei actually migrate to the rhizoids, and the sporangia will form multiple cysts. And once they're really matured, each one of the cysts will be containing hundreds, literally over 100 to 200 of small zoospores, which will eventually be released from the sporangia, and as the cell wall dissolves, and that completes the life cycle. And on the left is just another short movie that will show of a somewhat mature sporangia that I took under a confocal microscope. So these, what you can see is that there are these small zoospores moving inside already in this sporangia. And they're basically just waiting for the cell wall to dissolve and be released into the environment. Okay, so now let's take a quick look at the diversity of anaerobic fungi. And this is a figure published very recently by people working in the Xuefeng group from Oklahoma State. And they were publishing this new genus they have discovered recently, Pheromyces. And the phylogeny of anaerobic fungi are generally identified using the internal transcribed spacer region number 1 or number 2. In our lab, we generally like to use both the region number 1, number 2, and also the 5.8S ribosomal RNA in between, so that we have a greater confidence. But what's been very common practice is just to look at the region 1 of the ITS. And what you can see here is that there are about 9 to 10, in this case, 10 different genuses of anaerobic fungi. Some of the early ones include Neocatastax, which was categorized as Catastax under Protozoa for a long time, and also Actinomyces, which are named to honor contribution of cotton orpin, Cecomyces, Untomyces, and then the Cochlomyces. This one, Buckfloweromyces, which was discovered in the whales. So they have these really difficult names to pronounce, since they're Welsh. And Pyromyces, Anoromyces, and then the Pectoromyces are also recently discovered. So you can see that the diversity is not very, very large for anaerobic fungi. But how do they look in the bigger picture compared to other members of the fungal kingdom? So this is a synthesis plot generated by Igor Grigoriev at the Joint Genome Institute. And you can see that Neocalamastics is at the bottom here as a very early branching phylum, and along with Chytridomycota. So in fact, these anaerobic fungi for a long time were considered as part of Chytridomycota because they have very similar life cycle that goes from zoospore, which develops into a sporangium and eventually released back into zoospore. And up here, you can see the familiar higher fungi that a lot of people work with from the both Basidiomycota and Ascomycota. And you can find your familiar Saccharomycotina, which is a subdivision, and then a lot of mushroom subdivision up here. But the main take home here is just that these anaerobic fungi are early branching in the kingdom of fungi. And also at the beginning, it took people a long time to accept that these are fungi because for a long time, people didn't think that fungi can be obligate anaerobes. So some recent work based on taking advantage of next generation sequencing has allowed us to learn more about these anaerobic fungi. So this is work done in the O'Malley lab, where they have sequenced the transcriptome of Pyromyces finis strain. And what you can see is that they contain a large and comprehensive array of biomass-degrading enzymes. And in this chart, what is shown is in blue are cellulases, and in the red are hemicellulases, and then in the black are other carbohydrate active enzymes, catalytic domains. And if we look at how much, if we place this onto the tree that we're just looking at, and look at and count the number of biomass degrading enzymes, the anaerobic fungi under the Neocalamastigo-Mycota phylum, they possess the largest number of biomass degrading enzymes, much more than some of the familiar organisms that, such as the Trichodoma reesii, which is here. They basically have a lot more cellulases, hemicellulases, and peptide degrading and lignin modifying enzymes. And recently, last year, the whole genome were also finally assembled for these anaerobic fungi. And that has been quite a challenge because these anaerobic fungi's genomes are extremely AT rich. So in fact, the GC content of a lot of them is about 15% to about 20%. And this poses a very large challenge for some of the sequencing techniques, such as the short read based Illumina sequencing. So what was done here was to couple the sequencing from the short read Illumina based platform with PacBio sequencing. And this improved the quality of the genome. And by looking into the genome, we found that basically the parts list of fungal cellulosomes, and they are very important in tethering the plant biomass-degrading enzymes together. And these cellulosomes are well known for as being produced by bacteria for a long time. They're these very large protein complexes that can be hundreds of kilodalton in size, and they generally compose of this scaffolding on the bottom, and then you will have a cohesion repeated multiple times on this scaffold. And on the cohesion, there will be a doctrine domain on top of it, which connects this scaffold to the catalytic part of the carbohydrate active enzymes such as cellulase and hemicellulase. So if we look at the products of these anaerobic fungi, they basically, if you just look at the monoculture, composed of formate, acetate, lactate, and then they also produce a moderate amount of ethanol. They have to produce CO2, and they also produce hydrogen. So as a strict anaerobe, they have adapted, and actually they don't possess mitochondria, and instead they possess hydrogenosome, which generates hydrogen. And the exact pathway of how hydrogen is being produced from hydrogenosomes in anaerobic fungi still remains to be elucidated. But what I would like to point out from this early study done by Baltrup and Mountford, which are another two people who have done a lot of early work, is that if you cultivate these these anaerobic fungi with methanogens, the amount of cellulose or biomass being degraded will be higher, and the production of hydrogen is completely gone. So all of the hydrogen, as well as formate, they were all channeled through, and these electron sink products were basically gone, and electrons were transferred to methane. And you can see that the amount of carbon dioxide, total amount of carbon dioxide that were produced were much higher as well, higher amount of acetate being produced, and lower amounts of ethanol and lactate being produced. So one question that I have been trying to address and I've been working on is, what is the transcriptional basis for this metabolic shift in co-culture of methanogens? So to do this, I basically just need to do a similar set of experiments and sequence them using RNA-seq. So this is an experimental setup that I have provided. So here on the top are fungal monocultures, and I've grown them in triplicates on 4 different types of substrates, including alfalfa stem, which is very rich in pectin, bagasse. So in case you're not familiar with that, that's just the fibrous part of sugarcane after the sugar extraction, and reed canary grass, which is an energy crop, and as well as an invasive species in North America, and xylem, which is just a polymer, pentose polymer. I also cultivated them in co-culture with methanogens similarly. So what you're seeing here is that they are all growing very robustly. And what I have done with them is to monitor their gas production for about 3 days, and also measure the composition of their gas produced, as well as saving the liquid media for analyzing the volatile fatty acids they have produced. In addition to that, at the end of the experiments, all of these biomass were harvested for DNA and RNA extraction, and this work is still ongoing. We're still waiting for sequencing to be done. So stay tuned, and we'll find out hopefully very soon. So in the end, in summary, I would like to summarize the first part of my talk, which is that anaerobic fungi play a very important role in recycling carbon biomass. So they are an essential, even though a minority part of gut microbiome of herbivorous animals, and they possess a large, comprehensive array of biomass-degrading enzymes. And now we also have a better idea about what fungal cellulosomes are made of. And finally, they definitely form a syntrophic relationship with the methanogenic archaea. And at the end of this, I would like to acknowledge everyone in Michelle O'Malley's lab and Dave Valentine's lab at UC Santa Barbara, who has been great support. I've been learning a tremendous amount from everybody, and also Mike Theodoro, our collaborator from the UK, and also the Joint Genomes Institute that has been providing sequencing support, and the Exceed clusters, which provided a lot of the computational support and all the different funding sources. Okay, so now I would like to take a moment and ask the question, of as we transition into the marine environment. So if we think about the fungal, a lot of the fungi's feeding strategy, they excrete the depolymerizing enzymes, and these enzymes are used to break down organic matter to produce simpler compounds for them, which they use osmosis to take back into their cell for growth. This may be advantageous in digestive tracts of herbivorous animals because it's a very dense soup in there. But how about in the marine environment for fungi? Right. So it seems that such type of lifestyle would be quite disadvantageous because there's a lot of physical disturbance in the water column, even in the coastal area that is more sheltered. It is also the same. So for this reason, in fact, fungi in marine environments have been overlooked for a long time. We have seen in the past several decades of this rising field of marine microbial ecology, but most of these studies have been focused on prokaryotes and the abundant bacteria and archaea. But very few efforts really have been directed towards marine microbial eukaryotes. So I would like to basically draw our attention to one of these eukaryotes, which are marine fungi. So in fact, marine fungi have been studied for a long time. As early as in the 1940s, there have been publications about yeasts in the sea. Willy Honk from Germany have done a lot of studies in the North Sea on marine fungi. And I will get to that in just a minute. So these marine fungi, they could be major decomposers of woody and herbaceous substrates in marine ecosystems, and probably more so in coastal environments where productivity is high. So here is just a diagram illustrating that there could be plants growing on land, and there are also algae in the surface, and all these can sink down to the bottom, to the sediments, the coastal sediments, where marine fungi could, along with their prokaryotic friends, to remineralize them and produce carbon dioxide. And there are also recent reports suggesting that fungi can also be important in recycling nitrogen. They could be important in generating nitrous oxide in marine environments. And another idea that was brought up a few years ago is this so-called mycoloop. So one of the paradigm knowledge in oceanography is this notion of biological pump, which is that the primary production in the surface ocean, in the water column, they generally sink down, and most of it will be remineralized in the water column, but a small portion of it will actually reach to the deep ocean, and hence bringing carbon from the surface ocean into the deeper ocean, where the residence time of carbon is much longer. But what sinks to the bottom are generally larger particles. And in the idea of microloop, fungi could potentially break down physically, as we mentioned before, these large organic particles. And these are specifically fungi from the group of the chytrid fungi. And these will break down the large organic particles into small ones, which make them, again, more accessible for marine bacteria and archaea. So this could have implications on the efficiency of the biological pump in marine environments. So I would like to focus on a couple of the knowledge gaps about marine fungi, biogeochemical impacts, because previously most of the endeavors studying marine fungi have been focusing on their ecology, and their biology, but we don't have much quantitative knowledge about the contribution of marine fungi to carbon and nitrogen cycling in marine environments. Now, I would like to ask these two questions, and in this talk, I'm just going to focus on the contribution of marine fungi to denitrification and nitrous oxide production in marine environments. So, again, I am by no means the first person to study marine fungi. There have been actually a number of important books that have been published on marine fungi, starting as early as in the 60s by Johnson and Sparrow, studying fungi in oceans and estuaries. And then there's the Kohlmeyer in 1979, Marine Mycology, which is a great treatise on the marine fungi. And then there are also a number of later books by Moss in the 80s, Hyde and Pointing in 2000, and a couple of recent publications on marine fungi. So I'm very lucky to be able to participate in this expedition to the Eastern Tropical North Pacific oxygen minimum zone. Actually, the year here is wrong. It should be 2018. So it was just this past year, a couple months ago, on the research vessel Sally Ride. And on this map, what you're seeing is a global map of oxygen concentration at their minimum concentration. And what we can see, I would like to highlight, is these three regions of major oxygen minimum zones in the Arabian Sea, in the Eastern Tropical North Pacific, and the Eastern Tropical South Pacific. And where we went to in March and April this year was one of the largest oxygen minimum zones in the world. And if you look at the oxygen distribution in the water column, you would typically find the oxygen depth profile illustrated on the right here. So the oxygen is consumed rapidly in the mixed layer and into the oxycline. And there's about 100 to from about 100 meters to somewhere between 400 meters and somewhere up down to 700 to 800 meters, where oxygen is completely zero. So just a quick note about the nitrogen cycle. So nitrogen is most abundant in the atmosphere in the form of nitrogen gas, and they could be fixed through nitrogen fixation into ammonium. And this ammonium in the ocean will undergo nitrification, which is the oxidation of ammonium to nitrite and then to nitrate. And in oxygen minimum zones, under anoxic conditions, or perhaps just hypoxic, these nitrate is actually reduced back to nitrous oxide. So I'm skipping a lot of intermediate steps right here, but this and eventually nitrogen gas. But this process is where fungi could contribute to the nitrogen cycle. In fact, most fungi have been found to possess the denitrifying genes. All the enzymes from nitrate reducing to nitrite, and then the nitrite reductase, and then to nitric oxide, and then the nitric oxide reductase to nitrous oxide. So all these enzymes have been found and discovered in most fungi that have been studied so far, and most of these were just in fungi from soil environments. However, very interestingly, most fungi that have been studied for this pathway from nitrate being reduced all the way to nitrous oxide, they actually lack the last step of denitrification, lack the enzyme to perform that, which is the nitrous oxide reductase. And in the ocean, in these oxygen minimum zones, denitrification is one of the major ways that nitrous oxide could be produced. Another way is as a byproduct of ammonium oxidation. And in addition, it has been found that some of the nitrifiers that oxidize ammonium to nitrite could also perform denitrification by reducing this nitrite to nitrous oxide. So what we did on the cruise was sampling the seawater from different depths using a CTD rosette. And what you see here, just in case you haven't been onto a research cruise before, these are just bottles to collect seawater from different depths. And as soon as this CTD rosette comes on board, everybody gets very busy taking water out of these bottles and start incubation. But after we channel the water from these Niskin bottles into serum bottles, where we'll perform the incubation. So I'm going to go over some of the experimental design that I have had to tease out the fungal contribution to nitrous oxide production. So the first one I have used is inspired by the fungal community studying soil fungal community. And this method has been used for quite a long time. And in fact, in soil environments, fungi are often not the minority. They could be just as abundant, if not more, compared to bacteria. And to distinguish their contribution, we could perform incubations of seawater and add nitrogen 15 labeled nitrate to them. And this, by measuring the amount of nitrous oxide produced in the head space, we can calculate and measure the amount of nitrous oxide produced from nitrate. And if we add a specific inhibitor, in this case, I have chosen bronopol as an antibacterial agent to inhibit bacterial activity. Presumably, what I have left is only nitrous oxide produced from fungal activity. And similarly, I could also add a fungal inhibitor. In this case, I've chosen cyclohexamide, and this should tell us the bacterial contribution of nitrous oxide being produced from nitrate. And to be thorough, I also included a control that I have added both bronopol and cyclohexamide. And in addition to this, as I mentioned before, in addition to nitrous oxide being produced from nitrate, there's also nitrous oxide being produced from ammonium in these environments. So I did another parallel incubation with the same piece of seawater and incubated it with 15 labeled ammonium. So ideally, with these numbers, I can just calculate the fungal contribution to the nitrous oxide production, which equals the fungal N2O from nitrate divided by the total nitrous oxide produced from nitrate and the total nitrous oxide produced from ammonia. However, this method has a lot of limitations because whenever inhibitors are used, it is uncertain whether there will be non-specific inhibition. For example, if I had added too much bronopol, the fungal community could be partially inhibited as well. And the same applies to cyclohexamide. If I had added too much cyclohexamide, I basically can inhibit the entire community. So in order to get around this, I have introduced treatments of very different levels of bronopol and cyclohexamide. But still, we do not know for sure how much non-specific inhibition there will be. And this would introduce a fair amount of error when we estimate fungal contribution of nitrous oxide in this case. So that's why I also performed another set of experiments that is inhibitor-free. But these will require high precision mass spec measurements of the triple oxygen isotope and the nitrogen isotopomer analysis of nitrous oxide. So for these, I simply incubate the seawater without adding anything as a control, and also incubate them with some atmospherically derived nitrate. So the idea behind this is that this atmospherically derived nitrate would have a oxygen that has a delta oxygen 17 signature that is different from that in the seawater. So this is inspired by work by Scott Wankel's group that was published last year. What they did is they went to a German coastal area and sampled the sediment there. And they used this method combining both isotopomer, so showing the site preference on the x-axis here, which is capable of distinguishing the contribution of nitrous oxide coming from denitrification, showing the left here with a very low site preference value, and ammonia oxidation on the right, showing a very high site preference value. However, if only the isotopomers are measured, it is difficult to distinguish the fungal contribution here. As you can see, the fungal denitrification and bacterial denitrification, they would have site preference values that are very similar to nitrification or nitrifier denitrification. So the way to get around this is to add another dimension of isotope analysis, in this case, oxygen 17 analysis. So if the incubations, if we have a set of incubations where we added atmospherically derived nitrate, the oxygen molecules in this nitrate will serve as a tracer. And because denitrification, because nitrous oxide derived from denitrification would have to derive their oxygen atoms from the nitrate, they will show a different signature in oxygen 17 if the nitrous oxide is produced via denitrification. But this allows us to distinguish between fungal denitrification, nitrification, nitrifier denitrification, and bacterial denitrification, ideally. And again, this really relies on the prior knowledge of what these signatures will look like. And that generally is derived from pure culture experiments. So in fact, what you're seeing here on the top right, the fungal denitrification signature, a lot of them were actually derived from Fusarium, which is a very common soil fungi, and their signature of denitrification has been looked at. But there's not really been marine fungi that has been examined for their isotopic signature here. So there's a lot of work to be done right there. But if this method could be successfully applied, a mass balance calculation allows us to back calculate the contribution of different processes to nitrous oxide production. So what I'm showing on the right here is published in Scott Wonka's paper last year, and they have shown that in this marine sediments, the fungal denitrification could contribute up to more than half of the nitrous oxide they have measured in their incubations. But when we work with seawater samples, there's a specific technical challenge for these samples, which is the very low amount of nitrous oxide in these samples. So that is something that I am still currently working on, hoping to find a solution to. So in addition to these incubations on the cruise, I have also performed seawater filtration for molecular analysis targeting the marine fungal community. So on the left, I'm showing sequential filtration using a peristaltic pump for size partitioning. I have filtered them through 20 micrometer and then 2 micrometer and finally 0.2 micrometer filters, hoping to separate some of these fungal communities. And then, but this step is rather slow. In addition to that, I have also performed fast filtration, in this case using a diaphragm pump directly from the Niskin bottle. So in order to filter, I've timed the amount of time from the bottle is closed at the target depth to this filtration is completed for 30 liters of seawater. And that can take as little as just 10 to 15 minutes. So these can potentially used for metatranscriptomics analysis. In addition to that, on the cruise, I also started cultivating and isolating some of the fungi. So these are some of the cultures that I can show. So some of them are more successful than others. So these are in the center, what you're looking at are filter papers that I have cut. And then I just put them onto an agar plate with made of defined salt medium and plus a little bit of cellulobios as a carbon source. And you can see that these pink colonies are popping up. And all of these were also supplemented with antibiotics to control for bacterial growth. On the left here, it's somewhat difficult to see, but you can see some of this yellow colonies, which is a different type of marine fungi, perhaps growing on this plate. And I also have liquid cultures, where pretty sure I got some marine yeast growing in them, because they look very similar to some of the previous published images. So in summary, marine fungi are major decomposers of woody and herbaceous substrates in marine ecosystems, and the microloop could decrease sinking flux of particles. And they may play an important role in marine nitrogen cycling, and that's all of these are a work in progress that I would like to focus on as I continue to work as a postdoc at UC Santa Barbara. The main question is just to quantify really the contribution of fungi to marine nitrogen and carbon cycling. So in the end, I would like to acknowledge Simon's Foundation for funding and also all the cruise participants on this research vessel Sally ride, including members from the Ward, Cassiati, Babin, Zhang labs, and also from the oxygen sensor group from Denmark, and also my lab mates on land from Dave Valentine's and Michelle O'Malley's lab. And with this, I'm happy to take any questions.
All right. Well, thanks, Nick.
Yes.
You can come back to us here. Uh-oh, this is very meta. You can stop sharing your screen so folks can see you. And
okay.
Come on back to us. If we need to flip back to a slide, I suppose we can. But
yeah,
hi, that was great. Thanks so much for taking the time to share your awesome work with us. This has been really fun. So if you are watching at home, tweet us your questions, and Nick would be happy to answer them. I'm going to check Twitter here briefly, but I'll also start us off with the questions. So what is one of the biggest problems you have when you're trying to culture them in terms of getting things started? Like I was wondering in terms of, your choice of antibiotic and those sorts of things. What types of challenges have you guys had in terms of trying to get specifically fungal growth as opposed to, all of the other bacterial archaeal critters that are hanging out there?
Are you talking about marine fungi or anaerobic fungi?
Well, in both systems, I'm curious.
In both, I see, yeah. So for anaerobic fungi, actually, the biggest challenge is just to figure out what they need and design a defined media. And that will really allow us to pin down what their oxotrophy, basically. What do they really need? And right now, we still don't have a very good idea. So whenever we try to cultivate them and isolate them, we have to supplement the media with rumen fluid, which is, it's just, nature's magic juice, right? So, yeah. And
rumen fluid that you're working with here at Santa Barbara?
Well, we actually don't get them from Santa Barbara. So there is a supplier from Idaho. There's a farm. Every time they ship us, they would tell us, "Oh, this is from the cow Charlie," But but I also have taken a few members of the group up to UC Davis, where they have very active dairy research, and I have sampled. Actually, I didn't include any photos of the fistulated cows in Davis, and that was quite an experience. But
yeah, this was my very first graduate student project was sampling the fistulated cow at Davis.
Oh wow! Okay. Yeah.
Yeah, that's awesome.
I see. Yeah. So, but yeah, so we are now looking for perhaps alternative sources of rumen fluid because from Idaho it's very far. We're hoping to get some local source. So.
Yeah, nice.
Yeah.
And so, what about for the marine fungi?
So for the marine fungi, I think the biggest challenge is that they are just very dilute, very, very dilute. Mm-hmm. So I literally have to filter through up to 100 liters of water to concentrate these organisms. Mm-hmm. If so, in the digestive tract of cows, they are the fungi are 4 orders of magnitude lower than bacteria abundance. And if we think about if it's comparable in marine environments, or even lower, and we know that there's maybe about 10 to the 6th of bacteria in 1 milliliter of seawater, then that means there's only less than 100 or even less of fungi, right, in there, much less. Yeah. So people have all of these anecdotal reports of seeing cells that are larger than bacteria when they do microscopy, but really there have not been a lot of attention being paid to them. So I think the biggest challenge is just they're very dilute. Yeah. So it's hard to capture them. Yeah.
Yeah. Well, interesting. Well, Twitter's pretty quiet out here, but let's see. Okay. We've got lots of folks retweeting, and I think plenty of folks that are planning to catch us later. But
all right, sounds good.
What are the things that you are most excited about from your sort of marine fungal research and sort of future directions for what you're hoping to get from the project?
I think one of the most exciting part is to really fill in the blanks about their quantitative contribution to the biogeochemical cycles, because there have been just this tremendous body of literature in the past, 50 to 60 years of people working on marine fungi, but most of them have been just focusing on what they are, who they are, really, and people know, and most of those are also focused on in coastal environments where there's a lot of woody biomass input. But what I'm hoping to show is that actually in the open ocean, they're also out there, and then there have been one profile that was published by Silvio Pantoja's group in Chile, and but all they could do was just doing a microscopy and just start where the fungal cell was caucafluor wide and try to count them. But I'm hoping to show that in the open ocean, they are also playing an important role and an active role in the biogeochemical cycles. But on the other hand, I'm also excited to show more. That in coastal environments, they are also very active. So and they actually contribute perhaps to a lot. Just how much of the carbon recycling or remineralization in marine environments is routed through fungi, right, as opposed to bacteria. So.
Yeah. Do you have any plans for looking, I know you said you were doing that sequential size filtration. Do you have any plans for looking specifically at their particle association and trying to get it sort of at that resolution? Or what are your plans for sort of thinking about that? Because I have this wonderful image of them like forming those rhizaria through particles, breaking them up. And I'm kind of wondering what your approach is to getting at some of that.
Right, yeah. So some of those, I think, if we want to show definitively, I think we still have to rely on some microscopy, you know. But those samples were collected, I think, just as a first thing in order to basically get a molecular analysis of them and just look at the community, you know, particle versus, you know, free living ones.
Yeah.
Yes.
Oh, that's exciting. Well, I'm sure looking forward to seeing what you find. And thanks so much for taking the time to tell us about your work and
absolutely
share all of your thoughts. So for those of you that are watching out there, maybe at a later date, don't be shy. Nick's recently joined Twitter, so you can find him at his Twitter handle, which is Peng/Xuefeng, and you'll be able to get a hold of him and tweet him your questions directly, or tweet us here at Microseminar and keep the conversation going.
Awesome.
So thanks so much for joining us, and we are signing off. Thanks, Nick.
All right. Thank you. Yeah. Bye, Lizzie.